This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 804, recorded on September 10th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despomier.
Hello, Vincent and everybody else. It's a spectacular day today. It's breezy. Temperature is in the 70s, low humidity. The perfect day for being outside. So why are we all indoors?
No, you can't record outside. It's too noisy. It is very nice. It's 23°C here and very not humid at all. Few puffy clouds. Lovely day.
Yes.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's pretty much the same here, 69 Fahrenheit, 21 Celsius.
Outstanding.
Scattered clouds. We're just entering the best season here in Western Massachusetts. It's autumn and starting to feel like it.
I would agree.
Our guest today is from Points South. She is at the University of South Florida, which is in St. Petersburg. Mya Breitbart, welcome to TWiV.
Hi, thanks for having me. It's 86 degrees and sunny and not too cloudy, and maybe we'll make it through the next hour without a massive thunderstorm.
So you told us a few minutes ago that you look out over the ocean?
I do. So one of the real joys of being a virologist or microbiologist in a marine science college is that we get great views. So we're right on the water. We've got easy access to ships, and it's beautiful.
Nice. Very nice. Well, I see the water of the Hudson River. How's that? Is that good?
It's sometimes it's good.
Although I don't study it. If Mya were here, she would probably be studying the Hudson River, but I don't do that.
That's got to be a rich virus discovery environment, right?
Yep.
Yeah, for sure.
Interestingly enough, the energy cycles for the Hudson River are dependent upon bacteria rather than the herbivores are eaten by the carnivores, and then the detritivores take over. This system works on bacteria, and that's really a stumper. It's one of the few large river systems that actually works that way. So we would really love you to come here. Probably lots of phages involved in all of this.
Exactly.
You've driven up a fjord lately.
Yeah, exactly.
Not late. I did grow up in New Jersey, so I've got lots of familiarity with your area. In South Brunswick. So kind of between Rutgers and Princeton.
Sure.
Oh, that's not too far from where I am now. I'm in Scotch Plains, which is near New Brunswick.
And I grew up in Dumont in northern New Jersey.
You're north. You're north New Jersey.
Yeah, north.
I wouldn't have
My dad
guessed.
worked in Fort Lee and Englewood Cliffs, that area.
I live in Fort Lee.
My grandparents lived in Fort Lee. Yeah.
Cool.
Wow.
That's absolutely cool.
Cool.
What a nice connection.
Well, that's a good start. You grew up in New Jersey. And where did you go to college?
I did my undergrad at Florida Institute of Technology in Melbourne, Florida, so on the East Coast, and chose it because I wanted to be a marine biologist really badly. And, a lot of schools, you don't get to declare a marine biology major right away. You kind of have to come in as a biology major and specialize later. And as a high schooler, I had no interest in that. I knew where I wanted to go. I wanted to do marine biology and went to Florida Tech but I had no interest in microbiology at that point. And then during my undergrad, I had, as everyone says, some amazing teachers, teaching assistants. And it was a basic microbiology course where you have to identify your unknowns. That completely got me hooked. Like that. Great. Mind candy, loved it. And my TA for that was a grad student who was working on isolating bacteria out of Antarctic sponges to find novel antimicrobials. And so I started volunteering with him on his research as a very clueless sophomore and, kind of kept on the marine micro train since then.
Nice.
Wow.
Did you ever get to Antarctica?
I did not. I'm a warm weather person. I think Antarctica looks great in pictures, but I personally have no desire to go there.
Nice. So then you went somewhere to do a PhD, I presume, right?
I did. So when I was when I was an undergrad, I applied for an RU, and I did an RU at Scripps Institution of Oceanography in Farouk Assam's lab. So very famous marine microbiologist, and he was an incredible mentor to me. And the postdoc that I ended up working with in his lab was Forrest Rower, who eventually became my PhD advisor at San Diego State University. So I've actually worked with him in one way or another since 1998.
Wow.
Yeah, we had Forrest on TWiV and TWiM. Alan, you were on that episode with Forrest.
I was on the TWiV episode with Forrest, yeah.
And then more recently, he came on to TWiM, I think something to do with wastewater. Maybe they were looking for SARS-CoV-2 in wastewater. I don't remember. Anyway, he's always fun to have on. He's very really good, but of course also funny, right?
I listened to the TWiV was about their environmental sampling for SARS-CoV-2. I remember that one.
Oh, that was a SARS-CoV-2 one. Maybe that was Alan. Did we have him on again? I
thought we had him on for an earlier one. Wasn't he? Was he on the Penn State?
Okay, he's been on twice. Episode? Yes, Penn State was 391, and then 732. Oh, that was a citizen science project with Maria Isabel Rojas. Remember, they got people to collect swabs and give them to them, and they look for SARS-CoV-2. Yeah, that was TWiV. So 2 TWiVs, not a TWiM. Sorry. I get just like some people get their kids mixed up, I get my podcasts.
Get your podcasts mixed up
here.
Totally understandable.
Yeah.
So, and I guess before that, just in the spirit of openness, and for any younger career scientists that are listening to this, I think we always tell it like it's this really straight path. But when I was finishing undergrad, I was really interested in both microbiology, and I'd kind of had a taste of phage biology and still in marine science. And there weren't a lot of marine microbiology grad programs at that point. And so I kind of applied all over the place. Some real like hardcore micro programs, hardcore oceanography programs. I actually started my PhD at the University of Hawaii in studying oceanography. And I spent a year there. I took all of my kind of core oceanography classes there. I learned a whole lot. And at the end of that year, I switched programs and then moved to San Diego State to do my PhD in cell and molecular biology. And, not to be too much, everything happens for a reason, because it certainly didn't feel that way at the time, but it gave me this really kind of disparate training. So I had background both in oceanography, which I think I wouldn't have got my current job in a marine science college without it. But then I also got, cell and molecular biology, microbiology training as well.
Mm-hmm.
And was that a planned switch, or was that okay?
No, it was advisor relationships and core research fit sort of switches, but it worked out well.
Yeah.
Interesting.
So after your postdoc, you went directly to South Florida?
Yeah, I didn't do a postdoc. So I was very fortunate. So as soon as I finished my PhD with Forrest at San Diego State, I came straight to the University of South Florida as an assistant professor in 2006. And I can't believe it's been 15 years, but it's been 15 years.
So did you go there as an oceanographer or a virus hunter? So you identify yourself now as a virus hunter, right? So when you first went there, were you already a virus hunter?
Yeah, I think I was, but I still, I definitely identified myself in my job application. I think still in my formal title, I'm a professor of biological oceanography in the College of Marine Science. So I'm one of the few microbiologists here. John Paul was here for many years, and he did a lot of marine virology, really important work on lysogeny and gene transfer in the oceans. And so he was here, but other than him, I was the only microbiologist, and he's now retired. And I am the only microbiologist here within our college, which is very interesting.
I'm sure. All right. So let's talk about some work. I have to say, so I've always wanted to get you on TWiV, especially when I think I heard you talk in Maryland at ASV, was it?
Yep.
And you said, focus is for losers, right?
That is our lab motto.
But I always forget. And then recently I was reading a review article, it's called Beyond Coronaviruses, right? The virus discoveries transforming biology. And they quoted you several times about finding viruses in spiny-backed orb weavers. So that's our first paper here I want to discuss. Prevalence of a vertically transmitted single-stranded DNA virus in spiny-backed orb weavers from Florida, USA. So I think I've read elsewhere that you look all over the place for viruses. So why did you look in spiny-backed orb weavers?
I have a lot of them in my backyard is the very, very scientific answer to that. So we had a large project, which we're still working on in various aspects. It was funded by the National Science Foundation by the Assembling Tree of Life program. And, whether or not you think viruses are alive, we convinced this program that they should consider including some virus research and trying to understand evolution and connection between different organisms. And we in particular wanted to look at this group of CRESS DNA viruses. So it's CRESS, which stands for circular rep encoding single-stranded DNA viruses. So the kind of, if you knew one of them, it would probably be like porcine circovirus. These are really small. They have single-stranded genomes, typically they can be as small as 2,000 bases, 2 genes, basically a replication protein, which we can usually identify based on kind of sequence similarity. And the other one is we presume to be a capsid. We often can't recognize it as such by homology. And so we started finding these viruses in around 2006. They started popping up in our metagenomes. I guess about 15 years now, kind of right when I was making my transition. And it happened because of sequencing methodology. So before then, we do a lot of viral metagenomics where we purify the viruses out of a sample, kind of extract all the nucleic acid, chop it up, sequence it, put it back together. Before that, we'd been using Sanger sequencing. And that was right when kind of the next generation 454 instruments and everything started to come online. And all of a sudden, instead of being able to start with a nanogram of DNA, we needed a microgram of DNA. And so, we were already filtering hundreds of liters of water to get enough DNA to work with. So scaling that up didn't seem very practical. And so we looked into the whole genome amplification sort of methods, like rolling circle amplification, so 529, Templiphy, and those actually bias towards small circular genomes. And we didn't know that when we started. It was kind of touted as this unbiased technique, but of course nothing is unbiased. So all of a sudden we start pulling up these genomes out of everywhere, seawater, sewage, plants, animals, you name it, we find them. No idea what their hosts are at all, right? So the blessing and the curse of viral metagenomics. But since they're small and circular, we have complete genomes assembled. We're able to verify them that they're complete by back-to-back PCR. But we have no idea, what they're doing out there. And so as part of that project, we proposed to look across a whole bunch of taxa. So basically to go out and discover these in diverse organisms. And we were really focused on invertebrates. So we did a ton of marine invertebrates. We did terrestrial ones. So that was part of what brought us to the spiders, but we've also done copepods and ctenophores and crabs and grasshoppers and ants. I mean, lots of termite, fungus farming termites, all sorts of different things. And so the spiders were just kind of a convenient, in Florida, we put these cages around our pool to keep the mosquitoes out because they're so vicious. And the spiders, I don't know how they get in there, but they love it, right? It's this perfect environment for them to latch on, grow, have babies, do the whole thing without being preyed on by anything because it can't get in the cage. And so we have at any given time dozens to 100, I would say, of these spiders. No matter how often we sample them or release them out into the world, they get there. And what's cool is we can see them go through their entire life cycle there. So we actually see the egg sacs, and you can see the eggs inside. You can see when the eggs get little spiderlings within them, and then you can see them release. And so we just kind of, initially we just ground them up and did our thing and purified our viruses and sequenced them. And we actually found 2 viruses and this was just starting in my backyard. And then we tested, individuals to kind of look at prevalence. So one virus was really highly prevalent, and that's the one that we believe is vertically transmitted. So we followed that one up a little bit more, and I'll come back to that. But then there was also another virus that we found really sporadically. And that one we believe is of dietary origin. And so this is another kind of problem with the take a bug, grind it up, and sequence it. You might get things that infect that bug, but you might also get something that's, on the outside of them, or something that they ingested. We actually use it dietarily all the time because, if you grind up mosquitoes and you sequence them, you can get viruses from like their blood meals, right? So we do that actually to look at vectors. But in this case, we wanted to know the spiders themselves. So we started doing mad scientist things like, testing individual legs to see where within the bodies we could detect the virus, and sampling the eggs and expanding out beyond my backyard to knock on neighbors' doors and, get their spiders. And we found these over a few years. It started off as an undergrad project, which was great for that student, became a first authored paper for her. And yeah, we're still really, really interested in this virus because we see it in the eggs, we see it in the spiderlings when they're hatching, and we don't see any negative effects on the spiders as far as we can tell.
Do you ever find any spiders that don't have these viruses in them?
Yeah, we do. So it's not in 100% of them. And I think it would be really interesting if I was, if I knew more about spider behaviors and things like that, to follow up and do comparisons. Are there any differences in behavior or life cycle parameters between infected and uninfected spiders?
How big are these spiders?
What?
Sorry. Go ahead.
Sorry, it was just a side comment. How big are the spiders?
How big are they? How big are the spiders?
Typically like a fingernail sort of size.
Do they have any fluorescent little dots on their backs by any chance?
They do.
See, I've seen a lot of them in the oak. What the heck is the name of that? It's a swamp. It's a cypress swamp.
Oh, like
near Sarasota. No. It's on the west coast.
Okefenokee.
The Okefenokee. Is that what it is?
I think so.
No, I'm not sure that's the name, but I saw lots of those.
There is an
spiders.
Okefenokee swamp.
I'm sure there is. That's not the one though. This is It's a spiral. I'll forget about it. I used to do some consulting in Sarasota, and so I used to go to this nature preserve just to observe wildlife. And every one of the rails where you could lean on and look over the edge, in the corners there would be a little spider, and it had these little fluorescent orange-red dots on the back. And I tried to get some close-ups with my macro lens, but I never did. But is that the one you're talking about?
It's not that one. So I know this one with the little orange spots. This more looks like a kind of like a crab. They call them crab spiders, but that's not really the right way. Okay. They have like red spines that stick up for them.
Very pretty.
Yeah, so I looked this up. This is Gasteracantha canicroformis that we're talking about, right?
Yep.
So it's kind of a spiked frisbee.
Exactly.
Carapace on the back.
I see.
And they have different color variants. They're spread circumtropical in the New World.
You mean cephalothorax, don't you?
He's a virologist. Cut him a break.
I think it's actually
no. His grandfather was an entomologist.
No. I think Dixon. I think it's actually the top of the abdomen. That's that.
Oh, okay.
That sort of thing.
So have you? Do you find them in the offspring? You were talking about seeing the spiders in the eggs. Did you ever test them and see if the parent is positive, if the offspring are too?
Yeah. And we do find them in the offspring, and we tried kind of, so you can sample the egg sac itself. So we've gotten kind of the spiderlings as they've emerged from the eggs, but haven't actually emerged from the sac. And we've also tested some eggs. We kind of tried to surface sterilize them to the best of our ability, and then check within the eggs. And we do find them there too. Although again, not 100%, but they seem to be kind of good and happy and healthy. And it wasn't that I was super interested in, these spiders in particular. We had a good captive population to study, but I love this story because I think it's kind of what we do, right? Like you can go anyone can go into their backyard and, count how many different types of species you see. And like, they're all probably have viruses.
Sure.
Nobody's looked, right?
Yeah.
So we don't know what's there.
Seek and ye shall find, right?
Absolutely.
And that's kind of a theme in your work. I was looking at your website, and I thought I saw the paper initially. I was like, oh, okay, hunting in spiders. Where she's probably an entomologist or something. And then I looked, and you're doing everything. You're looking at environmental DNA. You're looking at fish. You're looking at just everything.
Yeah, we take the focus is for losers thing a little bit to heart. Although, I always say it tongue in cheek, but the reality is I'm also extremely jealous of people that have, study systems that they know inside and out, and cell lines and mutants and, tools for manipulating, because you can't quite get as far this way.
You don't have transgenic spiders yet?
No. Actually, I should have made that my pandemic project.
Right.
What could have gone wrong?
So you call these the first vertically transmitted virus in spiders, right? At least as far as we found, right?
Yep, to the best of our knowledge. But I think a lot of it is I think a lot of times the first is not because it's rare or, anything like that. It's just that we haven't looked.
Yeah.
Now
the other one, or maybe it's the one that's mainly in the trunk, which you think might be in it. Do you know what that's infecting? Is not infecting the spider? Is it infecting something it ate, or you don't know?
We think it's infecting something it ate because we also have detected it in other insects, like that and other species of spiders that we've detected, whereas we never found, this one that we think is vertically transmitted. We never detected that in anything else that we tested.
Okay. So you call these circularis viruses, right?
Yes, we just made that up.
And what is that based on? Because they're crest DNA viruses, right? The larger grouping, but circularis virus would be what?
Would be virus taxonomy. It's a mess. So when we first kind of started calling all of these crest DNA viruses, there just wasn't a term at all. And so you have kind of established families like the Circoviridae or the Genomoviridae, which didn't exist back then, the Gemini viridae. And the problem with a lot of these crest DNA viruses was that they share this kind of rep in common that we can identify, but otherwise they can be really different. So, we needed kind of a lump to like throw these all under to say they share these similar characteristics, but we're not really sure kind of evolutionarily how they're connected. And crest DNA became that. And then it became, I guess it was just last year maybe that it was designated as an order. Mm-hmm. Is that right? Let me double check that.
And you didn't want to call it St. Petersburg virus.
Well, I think we would have a lot of those by now. But yes, hopefully we've learned, it's a phylum, the crest DNA Viricoda. But that was just established in 2020. And a lot of things underneath that don't have designations yet. So for example, circularis viruses, there's essentially, there are a whole lot of names for things that mean either small or circular or some combination of the both in various languages. Mm-hmm. And our only other choice would be to call it, just virus 1, 2, 3, 4, 5, which doesn't give any information about what it is.
Or alpha, beta, gamma, delta.
You wouldn't dare do that.
No, who would do that? Who would ever do something like that?
I don't understand.
No, that would just never happen in virology. No. And so crest DNA viruses are infecting everything, probably, right?
Probably. We haven't seen and there's some structure within that. So, for example, I mentioned the Gemini viruses. Those are plant viruses, and they're kind of clearly plant viruses. There's a lot. The vast majority of these viruses we only know from sequence data, and so we don't actually have linkages to hosts on them.
Now, of course, in your case,
we find them all over the place.
A lot of this is just a detection issue, I gather, because they're single-stranded and small. We haven't seen them because we haven't had the tools to look for them, right?
Yeah, so a lot of even the early metagenomics techniques really biased towards double-stranded DNA viruses, or sometimes people specifically target RNA viruses. We don't have great ways of getting all the viruses together, and I think we only really pulled up these single-stranded viruses because we were inadvertently biasing our techniques. And not only did we find these crest DNA viruses, which infect eukaryotes, but we've also found just a huge treasure trove of single-stranded DNA phage. So these are in the Microviridae family, which is very famous because of Phi-X174. But actually, we don't find the Phi-X174-like microviruses out in the environment very much. We instead find these Gakusho viruses, which is Japanese for very small. And the known ones are like Delavibrio phage, MH2K, Spiroplasma phage, the chlamydia phages. So all ones that are known to infect intracellular parasites. But we see these viruses all over the place in various different environments, and we don't know the host for any of them, basically. We've also are really challenged in even trying to quantify them. And that's something our lab's kind of working on now, but more questions than answers. You know, we have thousands and thousands of genomes and we know nothing about their biology.
Well, more questions than answers is the whole point, right?
Career security, right?
Yeah.
That's right. That's right.
So is it fair to say that most of your discovery is for DNA viruses?
We've had to make choices, usually, and that's primarily, I think money driven. But the RNA viruses are also really fascinating to us. We've been one of the projects I'm really excited about right now is there's so much that's known about plant viruses and RNA viruses in plants, and the ocean is also full of plants, but they haven't gotten the same level of attention from a virology perspective because, it's not damaging crops and causing economic or agricultural problems. And so we started looking in seagrass, which are really important. They're important for carbon sequestration, for photosynthesis in the oceans, for habitats, fish nurseries, lots of good things, shoreline stabilization. And they're really, up until a few years ago, there weren't any viruses known in them. We went out into turtle grass, which is the Lassia testudinum. It's the main kind of seagrass in Florida, the climax species, and nobody had looked for viruses in it before. So there we targeted RNA viruses because, again, we were kind of thinking plants and lots of RNA viruses. And we found a Potex virus. So this is an RNA virus that's in the Tymovirales group, and it's similar to potato virus X and a foxtail mosaic virus X. Because of that, we called it turtlegrass virus X. So if we can, we follow kind of the nomenclature within a field. But what I think was interesting about this was we found a virus that was most similar to those in terrestrial plants. So from an evolutionary perspective, that's really interesting because plants started out in the oceans and then made their way onto land, and seagrass then came back. So, kind of when did they acquire this virus? And it was pretty different from these known viruses, but not any more different than different terrestrial Potex viruses are from each other. And so that's one that we're kind of following up on now, because same situation, we went looking for it in healthy seagrass, because we're really interested in just understanding not just the virome from a perspective of disease, but getting baselines and understanding what diversity is out there, even thinking about beneficial viruses, right? Or more mutualistic relationships. And we find this virus, this one was only in a really localized spot. So where we first went looking for it is the only place that we can find it, but we find it there for years after years. So it seems stable there. And our next steps now that we're trying to get at are understanding, and what the heck is this virus doing out there in the host? Is it conferring any benefits or is it detrimental at all? How would that be affected by something like a red tide or increasing temperatures or changes in shoreline conditions? And we don't know any of those answers, but there's of course such a wealth of plant virology knowledge and research that hopefully we can benefit from, even though we don't quite have as many tools in seagrass that we do in, Arabidopsis or something. Right.
Have you thought of looking in Chlorella by any chance?
In Chlorella?
Chlorella. The algae that are mutualistic with corals.
Oh, like Zooxanthellae.
Yes.
Yes. So I have not done any work with that, but there are lots of groups. Adrian Correa in Texas is actually doing a lot of work looking at these algal viruses that live in symbiosis with corals.
Right.
So they're dinoflagellate viruses.
Really?
I'm not doing any of that work, but we do have a project right now. So the organism that causes red tide here in Florida, which is really nasty and devastating, is also a dinoflagellate. It's Mya Brevis. And we're right now actually collecting samples to try to do some virus discovery in those, especially kind of during the termination stage of blooms. So when we see the blooms kind of start to die and decline, to see if we can identify any viruses, and then, either, I guess, as a baseline, gain a better understanding of what's regulating these natural bloom cycles, but then also, potentially down the line as a possible mitigation strategy. We're not quite at that stage yet, but it's interesting to think about.
Right. To think that a university actually named their football team after red tide.
Is that
Alabama
right?
Really? Maybe they should name another football team Interferon or something to counteract it.
At least it's not going to be changed.
No, it won't.
Because there are other names that have to be changed, as you know.
Indeed.
So I wanted to just briefly highlight some of your other works here, just to give listeners a sense. And one of the papers is, which you touched on, which I think is the parent project of the spider work, virus discovery in all three major lineages of terrestrial arthropods. And here again, single-stranded DNA viruses, right? CRES.
We were specifically targeting that group of viruses. So we didn't look at RNA viruses or anything else.
Right. So you captured each kind, right? Again, around Florida somewhere?
Yep. So this was all just, the majority of it was opportunistic sampling based on what we could see and what we could identify. But we also worked with a lot of other researchers that helped give us access to different groups that they study.
Mm-hmm. And you find CRES DNA virus because that's what you're looking for, as you say. And so does a spider have a different one from an insect than a millipede? Are they all different?
They are all different. So we haven't really gotten, at least when we do the discovery, we're not kind of picking up the same thing all over the place. So it does suggest that there's, kind of species specific relationships here. But the CRES DNA viruses in general, they don't kind of nicely fall out by host, right? So that we can just kind of make inferences about what they infect. So we don't just have like one spider cluster that's different than the millipede cluster.
Okay. Yeah, that would be easy, right? If it were that way. Yeah.
So much easier.
And so you can't say anything about how they're transmitted either, right? If they're vectored in some way, I guess. Is there anything that bites a millipede, Dixon?
Hard to say. Millipedes don't have biting mouth parts for the most part.
Would a mosquito bite a millipede? I guess.
Hardly. I don't think so.
Okay. So
first of all, mosquitoes don't feed on blood, so you remember that. So they feed on plant juices.
Yeah.
So that's not part of their food system. It's really very interesting to see if though any of the insects that the spiders eat get their viruses maybe from plants. Do we know?
So we've done this a little bit with dragonflies. We did mosquitoes, and then we did a lot of work with whiteflies because they're vectors for a lot of plant pathogens. And then we kind of got the idea. We were working with this is Karina Rosario's work with my group, and then we were working with Arvind Varsani, who's now in Arizona, and we're kind of got this idea to look at dragonflies because dragonflies are they call them the vacuum cleaners of the skies, right? They're voracious feeders and flyers. They eat anything that flies. And in those we can detect things that, maybe are infecting the dragonfly. We're very careful with the name and we try to do a lot of dragonfly associated because we really can't prove it a lot of times, but we see things that they ate. So from other insects. And then we've even detected plant viruses in the dragonflies. So that's kind of probably, it's like the Russian nesting dolls, right? We see what they ate and what something they ate.
Sure. Very cool.
Yeah. Well, there are also arthropods that parasitize arthropods. I'm thinking of Varroa mites on bees, but I know there are a bunch of other mites that little tiny.
In the oceans, people have suggested looking at naphid isopods, so these kind of parasites. And I think there's there's a million things that we could do. And we still definitely get pulled in a lot of different, we should look in this because nobody's ever looked in it before. I think there's a huge amount of value, obviously, in kind of serving natural diversity and just understanding the gene pool that's out there, how it's differentiated. But also, in order to kind of take some of these findings to the next level, I suppose we are going to need to choose one or more model systems to devote the rest of our lives to. And that's where I've never... How do you make that decision? I ask virologists this all the time, actually, when I say, like, how did you decide that you're going to work on polio? Or how did you, decide that you want to study this species? And I really struggle. I struggle with that a lot.
It's biophilia. I worked on a worm that makes a spiral inside of a muscle cell. And I was just absolutely stunned by the symmetry of this organism. And I stayed with it for... Well, I'm still alive, so I guess I can say I can still think about it. But I was charmed by this parasite of all things, and to think it can live for 30 years inside of a muscle cell. How does it do that? So those are, lots of questions. But I wanted to. I'm sorry, this is not about me. This is about you. I wanted to know whether or not you've ever thought of getting samples from thermal vents, from animals that live in thermal vents, and see whether or not there are viruses that can survive at those temperatures.
Yeah, so there absolutely are. So there's a lot of researchers that have looked at viral communities in hydrothermal vents, and they're absolutely there. They see a lot of lysogeny actually in the phage communities. We sampled a few deep sea critters, including the Riftia worm from hydrothermal vents. For this project, we didn't find Racaniello viruses in them, but we sampled 2 specimens. Those are pretty precious specimens, and we have to destructively sample them to do our work. So beggars can't be choosers. But absolutely, I mean, they're absolutely everywhere. And I think sometimes we stumble into them without even meaning to. Like, obviously, if we decide to sample a spider or crab or whatever, that's a decision we've made. But I know you guys have talked on the show several times about the plant viruses in human feces, and one of my favorites, the pepper mild mottle virus. And that was one I never really thought about plant viruses. So I started my PhD with Forrest. I was working on seawater and seawater viral communities. I got to sample, by going to the pier, like hanging out on the beach, and it was all beautiful. And then you guys know Forrest, so I'm sure you can see this unfolding in your mind. One day he just came into the lab and said, "I think we need to study viruses in human feces." And I was already hooked by that point. I couldn't say no. There was nothing we could do. And then all of a sudden, instead of, sampling days at the beach, I'm going around getting a reputation in my program for asking different grad students and faculty to give me samples in Ziploc bags that we could process.
There's a metaphor here. He lured you with the sunset at the beach, and then
This is true.
Absolutely. Please give him crap about that the next time you
It's not a Pinot Colada that she was after.
Exactly. But then that project was kind of started. We were working with the Genome Institute in Singapore, and they were really interested in finding human viruses, and, thought by sampling the fecal samples, we'd find all these human viruses, and we did our metagenomics, and then it was just completely dominated by plant viruses. And so it wasn't, all of a sudden all our sequence reads are pepper virus. Okay, what's going on? Is it contamination? Whatever. And long story short, we were able to kind of trace it back to a dietary origin. So we eat a lot of these pepper viruses. They're super stable. We get them in sauces and spices, all sorts. One of the most fun things we got to do was in Singapore, actually going around the Genomics Institute food court, and we ordered one item from every vendor in there. PCR'd up this pepper virus. You can find it absolutely everywhere. And my lab's been using the pepper virus as kind of an indicator of wastewater pollution and looking at human health impacts from that. But one thing I wasn't involved in at all, but have been really just excited to see pop up over the last 18 months is so many people who are monitoring for SARS-CoV-2 and wastewater using the pepper virus as kind of a control, right? For knowing things are working and normalizing quantities. So you don't always know where it's going. And I love that.
Well, that's the beauty.
So if you backing up to this idea that you need to settle down on a model organism, I mean, I'm looking at your body of work, and I'm not sure that you do. Because there
I will be
aren't a lot of
listing you as a
people-
reviewer for my future proposals, Alan.
There aren't a lot of people who are doing this kind of broad-based, let's just go look for stuff.
Right. That's exactly
And
right.
it seems to me, at least, and I don't really control the purse strings on anything, but it seems like that's in itself a useful thing.
Absolutely.
Yeah, I agree. And I would never give up that side of it. I love the discovery side of it. I love the unexpected and all the questions. I do get frustrated with our, ability to take it to the next level, right? So, and kind of how you get beyond the sequence into the ecology. And to do that, we probably will have to pick a few that we want to study more deeply or get some eager collaborators and work on that.
I mean, you could theoretically, based on what you've told us, these images of big networks always pop up in my mind. If there were specific viruses which survived but didn't infect, you could actually find out who's eating who in a food chain or a food web by simply looking at the kinds of viruses found in the gut contents of these carnivores, herbivores, etc., omnivores. It's an ecology that's waiting to happen, it sounds like to me.
Yeah, absolutely. Especially if you start combining with that, like Vincent talked about the environmental DNA, if we, maybe that was Alan, sorry, but if we kind of combine these different techniques, right? That's like you can look at the viruses, we could also look at, what other types of host DNA do you detect there? And even though those projects, we have some projects that aren't, virological at all. Like we're using DNA sequencing to identify fish egg species composition and understand where fish are spawning. So,
absolutely,
maybe for a different podcast, but I think they're all just applying these different methods to like get new ecological insight, which is really fun. And those are projects I mostly started because I'm a lonely molecular biologist in a marine science college. And so I saw, I want to have friends and colleagues. And we have a lot of people who work on fish and, benthic ecology and lots of other systems. And so that was a great way to combine skill sets.
Is there a annual meeting that attracts marine virology or marine microbiology that excludes all the others so that you can meet people of similar minds?
Yeah, there are many. So there's an aquatic virus workshop that's every other year. It actually just happened in July, and it was in Japan, which unfortunately I participated, everyone participated via Zoom on their computers. I had been looking forward to visiting Japan. I have never been, but so that's a group that really started out focusing on algal viruses, but has gotten wider. If you kind of get beyond the marine realm, there's viruses of microbes conferences, but then, we don't have good, I guess, environmental virology conferences. And I don't feel like I have a home meeting kind of in the way that like, I talk a lot of virologists, ASV is their meeting, right? And they go every year. I've been twice, once on my honeymoon. My husband likes that story. And we're still married despite the fact that I gave a talk within 24 hours of our wedding.
Oh, wow.
We honeymooned in Canada. Was when the meeting was in Canada. And so we did that. And then the one in Maryland. And it's fascinating for me, but it's also kind of not quite my people. Like I don't know, how interested your average virologist is in a spider virus or whatever, but we also wouldn't fit in an insect meeting. I do a lot with the American Society for Microbiology, but that's also an offshoot, or the ISME, the International Society for Microbial Ecology. So I guess I have many homes, but maybe no place of real belonging.
Sure. Yeah. Sounds like a Gordon conference to me.
Yes.
It'd be a perfect topic for that. Have you thought as to whether viruses, let's say in the marine environment, if you want to take that for an example, where you've got filter feeders that depend on microbial life for their livelihood, and viruses, of course, intercept the microbes first in terms of phages, et cetera, and limit. Do you think viruses are limiting certain filter feeding populations in natural settings?
Yeah, it's a good question. I don't think so, because I tend to think of the viral infection in the oceans as kind of fueling this marine microbial loop of kind of this recycling center, like what you were talking about with the Hudson River and the, bacteria really being the base of the food chain. So we have a lot of bacteria, a lot of very active bacteria in the marine environment. And then, each of those bacteria can be infected by phage, and that would lyse them, produce more viruses. That just releases more dissolved organic matter, which can then be taken up by the bacteria. And, so I think of it as like just fueling this rapid cycling. There hasn't been a ton of work into some of these filter feeders may be able to, or may capture viruses through their filter feeding. It depends on the exact kind of apparatus as well as the size of the virus, because we know there's certainly giant viruses, right, that would be captured by anything that's catching bacteria. And we do see viruses, like when we use the pepper virus as kind of a tracer for wastewater, you can track them into shellfish, for example, right, which, well-known filter feeders. So I don't think it's a detriment at all, but I think it's just a key part of the nutrient cycling.
Right. A lot of that is totally not understood at all, isn't it? I mean, how much protein is released per bacterium, for instance, that then generates more life because of the nutrients that it's created? I don't think about this a lot, but when I do, I tend to obsess on it.
Yeah, we do a lot of multiplying very small numbers by very, very large numbers, right?
Exactly.
Because, we may be measured in our 1 liter bucket of seawater, and then we multiply by the volume of the oceans. But they're certainly releasing a lot of material, and that kind of lysate, the bacteria guts that gets spilled out, is highly bioavailable. That can be taken up really quickly.
Amazing.
And in general, we don't think very much about the virus particle itself outside of the host, right? So when you talk about phage, people always talk about lysing the host or changing the host metabolism, and affecting it, horizontal gene transfer, things like that. A virus particle just kind of floating out there, a virus particle contains carbon, it contains nitrogen, it contains phosphorus.
That's right.
Those are all parts of these dissolved pools. It is good eating. And we've been working a lot. One of the projects I'm really excited about right now is looking at the amount of iron that might be complex within phage tails. Because there's some evidence from E. coli phage that they can have iron as part of their structures. And iron is a really important limiting nutrient in the oceans. Not enough of it, you don't get phytoplankton growth. And so we want to now kind of
red tides.
Absolutely. Yeah. So we're interested in seeing, characterizing what different trace metals and other things are part of these virus particles and then are contributing to the dissolved pool, which we think of as just being dissolved and there for uptake. But a virus particle is clearly not dissolved despite being in that size range.
Yeah. So you also have this model that I liked the name of, the Trojan horse, where the viruses are complexing iron possibly as a mechanism of gaining entry into bacteria that are hungry for iron, and so they eat the iron. But there's a virus attached to it.
Yeah, and we don't know if the Trojan horse or the Trojan horse part of that happens, but we do know that some viruses have iron in their tails, and we do know that some phage use siderophore bound iron uptake receptors as their infection receptors. So phage can actually compete with siderophores for access to that uptake site. And so if those two pieces kind of happen at the same time, it's really interesting. And then we're kind of further hypothesizing that when the new phage they assemble within the bacterial cell. So we think that's kind of the most likely place that they're getting their iron from is within the bacterial cell. We're trying to trace that with labeled iron. And then the idea is that when that cell lyses, instead of all of that organic matter being available for uptake, for example, some of the iron may actually be contained within the virus particles. And so that would really affect how we model kind of biogeochemistry in the oceans and cycling of these nutrients.
So if there's a million virus particles per teaspoon of seawater, right? So if a filter feeder takes in a lot, could those particles be nutritious?
Yeah.
I mean, they're
these are tasty morsels, right?
What's your favorite low-calorie, high-protein drink? Viruses. Yeah.
Yeah. I mean, they may be small, but if you eat enough chocolate chips, you're going to gain weight. I think, and they have distinct kind of CNP ratios, right? Compared to other things. So the phosphorus in particular might be really important in certain areas of the ocean. I think you're going to have different important factors in different places, but I think we need to start thinking a little bit more about these particles that are out there and how they might be contributing when they're not infecting.
I mean, the problem is that
C's and P's,
many of these particles are highly stable, and that's how they, survive, retain infectivity in the environment. So they may be resistant to stomachs of various filter feeders. I don't know, but people always ask me about that.
Possible.
Yeah.
If you took a core sample of the ocean, I can't think of another way to put that, and did a microbial analysis, let's say per centimeter or per foot or maybe meter, in some cases to find enough to make a decent count out of it. The virus particles, as far as I understand, stay up in the photosphere. Is that correct?
No. So,
they filter down. They actually fall down as part of the snow that goes down to the bottom.
It depends. I guess it's not a super straightforward. They're small enough that they wouldn't just be sinking on their own. But they can be sticky. And so, and even, as we were talking about the recycling, that concept is called the viral shunt. This idea, right, that the viruses infect and then that creates more dissolved organic matter and fuels that loop. But there's also a kind of, I don't know if it's really a competing hypothesis, because I think both are happening, about the viral shuttle, being the idea that as you produce all these sticky lysates and all these viruses, things might actually clump together and then accelerate settling and kind of contribute to the biological pump or the sinking of carbon,
cycle,
that way.
Gotcha.
So they definitely do end up on the seafloor. And if you were to take a sediment core, you can, just like people use sediment cores for, we can look back into kind of past viral communities. I think you have the extra caveat of stability, right? Part of the reason I think, we always find the pepper virus and we don't, as opposed to kind of other viruses in our diet, is that is just a super stable virus. You can burn it, you can heat it, you can hit it with all sorts of enzymes and and it still survive, or it still at least remains intact, right? And you can detect its RNA. It's detectable. And so I would imagine that as you go down through the sediment, unless you're in a very high preservation environment, you are going to have preferential decay of certain viruses and others will get retained. But it's a really interesting idea to look into.
Yeah. One of these global ocean surveys, they did sort of that sampling at different depths. And you can find all these data online. I'm sure you know about this. And Steve Wilhelm first showed this to me. And you can see a gradient of viruses as you move out of the photic zone and you still find them deeper, but they're much less, right? So in a way, Dixon, that's been done by sampling different depths. Okay. Right, and then doing filters.
The other thing to think about, to think about is the diurnal migration of life from the depths up to the surface, particularly at night. And they may be seeding the upper levels of the oceans with the things that, participate in cloud formation and stuff that's really important. That if you don't know that and you start destroying it, you're going to be real sorry very soon. And what you're doing is absolutely critical to knowing whether that's going to happen or not. I don't know if you've looked at E. Huxley or not for a host. But,
I haven't actually, but a lot. I mean, that's a huge field of research. There's a lot of people working on that one. But I think, the vertical migration gets also really interesting when you consider the oceans aren't, they're not static, right? There's currents and there's different currents at the surface that there are deeper. So for example, when we've looked at copepod viruses and, even if you think about copepods that eat E. hux or eat other phytoplankton, and you could see that you might have a bloom here and then it gets ingested and then the copepod, migrates during the day. And then when it kind of comes back up, it's coming up into a different patch of water, right? Exactly. So the phytoplankton that were at the surface, maybe they've moved on and now they're entering a naive population that could be infected by those viruses, assuming the viruses survive and several million other assumptions, but I think these are all really interesting when you think about how things move around in the oceans and the virus introduction just naturally. Plus, then you can add things like ballast water and human migration and inputs on top of that, and it's complicated.
Yep, and don't forget whale feces. I mean, that's a real distributor, if a potential distributor, right? So it's I must say that when I was growing up, back in the days when I was trying to decide what to do, I did have an opportunity to become a marine biologist. But the caveat there was that I had to know how to fix an outboard motor. And I didn't know how to do that. And so instead of going to Bimini and becoming whoever I would have become, I became this. Well, the guy who took that job didn't know from Shinola about how to fix an outboard motor either, but he just lied about it and said, sure, I know how to fix an outboard motor. And, why the hell didn't I think of that?
I'm not advocating for lying, but there's a lot of on-the-job learning, I think, with any of these positions that you take.
No question.
Absolutely. And I can
for reference,
with the best of them.
Most people learn how to fix an outboard motor because they have to. And the breaks. That's something you pick up when it happens.
Yeah.
Well, these two couple that I would have worked with were studying dolphin language, so they were venturing out into the depths of knowledge as well as to the ocean.
So, Maya, let me ask you two final questions. Do you have a favorite virus discovery story that was particularly fun?
It's like asking somebody to choose their favorite kid. I think for me it was really the pepper virus, just because everything about the story was pretty funny and pretty unexpected. My favorite failed one that you'll never see in the literature because we never got it to work was that after, in case it wasn't, enough that Forrest was torturing me with human feces, he thought it would be really cool to look in cow rumen. And I don't know if you know, but he grew up in Idaho and his parents run a farm in Idaho. So he had his father ship us multiple kind of milk cartons full of cow rumen fluid. And I was alone in the lab because your lab mates will never forgive you if you do this during the day. And I put some balm on my nose so that I wouldn't smell it as badly. And I set up with my tangential flow filter system and I was wearing proper safety gear and everything, but it clogged almost immediately. All of the tubing flew off the filter and I was covered in cow rumen from head to toe. And needless to say, I never published a paper about viruses in cow rumen. So for any student out there who thinks their life is particularly bad right now, at least you're not covered in cow rumen.
Wow, that's good. Good one. All right, one more. If you hadn't become a scientist, what would you have done?
That's such a great question. I still think about this all the time. I guess it's all connected to science though, but more on the environmental side. So I was a Girl Scout when I was growing up, and I've continued to be involved in Girl Scouting throughout my life in various roles. And for me, that was just absolutely critical for kind of exposing me to potential careers and getting me excited about science and the environment. And I absolutely love education and outreach. It's a really important part of my research program, my research group, just showing kids that these opportunities exist and, kind of seeing that look on someone's face when you tell them that they have millions of viruses in their body, and then you can explain that not all viruses are bad and stuff. And I know that's kind of linked to science, but I really think that the education and the, getting young people excited, getting everyone excited about their environment and this idea that you could find something brand new. I think so many people, especially because of the way that we teach, we teach knowledge, we teach what is known. We don't spend nearly as much time talking about what is not known. And the idea that you could just walk into your backyard and find something brand new, that's really exciting. I think it's exciting no matter how old you are. And so kind of dispelling this idea that all the cool big discoveries have already been made, no way. There's so much more fun ahead.
Yeah. Yeah, when I was a kid, I had a book called Science in Your Backyard. I loved it.
That sounds great.
Actually, I stumbled across it the other day. I have to make it a pick one of these days because it was just, I was just amazed. This is in my backyard. Wow. What I didn't do is go out and find it. I should have done that. All right. I guess
not too late, Vincent. There's a lot of science still in your backyard.
Well, I've always wanted to trap mice in my backyard and look for some viruses. And I found out recently that mice are protected. You have to get a license to capture them.
Really?
They're owned by the U.S. government, right? Like the mineral rights. The wildlife belongs to the state or whatever. But anyway.
This is part of the reason that I like working with invertebrates.
Yes, you don't have to get a license.
Less permits.
Vincent, you need to scale down to spiders.
When I see spiders, I see all kinds of beautiful spiders and insects all over, and I always wonder what's in it. But that's Maya's job. That's not mine. Although I tell you, last night I trapped a mouse. We had some idea that there was a mouse upstairs, and because it was eating all the bird seed, something was eating all the bird seed. So I got up this morning, and there it was. It was a deer mouse, quite clearly.
A deer mouse.
And
Permiscus.
Permiscus maniculatus, probably, maybe not. But I came in, I told Amy, my lab associate, and she said, "Did you bring it in so we could sequence?" I said, well, no, I figured it was dead for so many hours. We're not going to, but next time. Anyway, Mya Breitbart is from the University of South Florida. Thank you so much, Mya. It's been a pleasure talking to you.
Thanks so much. This was really fun.
And thank you
fantastic.
Thank you for wearing a great headphone set that makes you sound great. Really appreciate it.
Yeah.
I was glad you mentioned that in your email. So for future people, thank you, because I do meetings in here all the time without headphones and nobody's really complained, but they're not being recorded for podcasts. So
right.
that was
your
good
shirt,
reminder.
Your shirt is the homage back to Hawaii, I presume.
I didn't think about that, but yeah, I loved Hawaii.
I'm sure.
That's somewhere I could see myself again, but I always feel like it's important to tell that part of the story because, so many people think that if you have one kind of misstep or an unplanned thing happen, that you're toast. And it certainly felt that way at the time.
Yeah, sure.
It worked out okay.
Exactly.
Thank you, Mya.
All right. Thanks, everybody. It was great. Thanks for inviting me. Bye.
Bye-bye.
Guys want to do some email?
Sure.
Sure, why not?
Dixon, can you take the first one?
I can. Yes. David writes, pre-comment, thanks for TWiV. In a recent TWiV, I listened to your discussion about global warming and our prospects. I got the idea that there was a categorical statement that the best we can do is slow down or stop the warming of the planet. We cannot turn the process around. I disagree with that assessment. Of course, what we need to do, we will unfortunately not do, simply because we, as a nation, lack the political will to tackle long-term problems until it's essentially too late. Okay, but what could we do? Yes, the planting of more trees. However, living here in the Northwest rainforest, we are witnessing the turning of years of lumber growth into CO2. Eating less beef would help too, but again, these are only mitigation strategies. The essential thing we need to do is to generate power. We need to generate lots of power. All of our solar panels and recycling are just tinkerings at the edges. We, the people of the world, need to generate more power than we consume so that we can begin to undo the years of damage. In reality, there are only two possible power sources for the scale of power that we need. These are fusion and fission. Despite recent advances in fusion, we are a long way off from that reality. Fission, on the other hand, is quite doable. With fission, we have three possible approaches, with each having a variety of nuances designed options. The three are burners, burning U-235. Nearly all reactors in operation today use this approach. Light water reactors, that's the continuation of that sentence. Light water reactors, boiling water reactors, fast breeders, turning U-238 into plutonium-239 and fissioning that isotope. There are a number of designs in the works, including one funded by the Gates Foundation, or possibly by Bill Gates without the foundation. Since the early 1970s, the U.S. has put a lot of dollars into research on fast breeders. For a variety of reasons, their potential remains unrealized. Thermal breeders. turning thorium-232 into uranium-233 and fissioning that isotope. Again, there are a number of designs in the works. To me, the most interesting is the LFTR, liquid fluoride thorium reactor. This reactor has the fissionable fuel in a liquid state, dissolved in a salt of lithium fluoride and beryllium fluoride. The fact that the fuel is in a liquid state makes it repurposing in a continuous manner possible. There are other benefits, including inherent safety, as long as the laws of physics continue to apply, a high heat byproduct making possible turning seawater into freshwater, or removing CO2 from seawater as a byproduct of the reaction. The U.S. never put much money into the thermal breeder program. Nonetheless, Oak Ridge ran one on a shoestring for a number of years without incident and gained a lot of knowledge on the subject. Suggestion. Listen to a number of YouTube videos by Kirk Sorensen, also one called Energy Cheaper Than Coal, which discusses the reality that the poor will use coal to save themselves unless we can provide a cheaper option. Of course, I am not suggesting that Kirk Sorensen is the only source of information. If you listen to some of these videos, you will go into source data, including a trove of old documents rescued from a closet at Oak Ridge and scanned to PDF for all to read. The upshot of my comment, we can save the world with nuclear power. Unfortunately, the people who believe in science don't believe in nuclear, since all they know about nuclear power is the light water reaction, and people who don't believe in science, well, no further comment required. We can save the world. Will anyone listen? Dave. That was a good letter.
I didn't know much of that, and it seems to me, I don't know anything about this, and it seems to me that people are afraid of nuclear power, right?
They are. That's the one thing that's right. They are.
Right. But the opposition to nuclear on the science-informed side is not entirely as irrational as Dave implies here. There are real issues, and a lot of them stem from the fact that you can use a lot of the byproducts of these reactors to blow stuff up.
Correct.
And so, this notion that, well, we could have everybody build more nuclear plants. You notice the term breeder in a lot of these reactor designs. That means they produce more fissionable material than they consume, which sounds great for an energy plant. The problem is that it is the perfect way to cover up an atomic weapons program. And we have systematically tried to prevent people from developing atomic weapons. With some good and some bad reasons. So there's a lot going on here that needs to be dealt with in a nuanced manner. That is not, this is not a slam dunk. No. This is not something we can just do, but it does all come back to humans are horrible. The problem is that we can't be trusted to responsibly manage these resources.
Yeah, we have met the enemy and they is us.
Yeah, very much so.
By the way, Alan, does your daughter like the two finalists in the U.S. Open, the two female finalists?
Yes, she's very excited about the fact that it's going to be an all-teenage
yeah, that must be cool, yeah.
Yeah.
I would guess as a tennis player herself, right?
Yeah. She was bummed Naomi Osaka got eliminated. We hope Naomi sorts out her various issues and gets back. But
Fernandez drove through a lot of well-ranked players.
Yes,
absolutely.
Yes. So it is no shame to have lost.
It was a remarkable match last night.
No, I mean Osaka was bummed, but I think she shouldn't be because the girl beat a lot of really.
The girl is just a powerhouse.
She's an incredible talent.
On fire in this tournament. Yeah,
exactly right. Exactly right.
Yeah.
Guy writes, interesting paper from Israel now reports Delta variant RT. That is the the version of the R0, the transmissibility index that you calculate after there's some immunity in the population, of less than 1.4 for all classes of patients, vaccinated and unvaccinated. Could this be a different Delta variant? Nope, just different behavior. No gigantic motorcycle rallies in Israel. All the media hype and medical pundits announcing RT of 6 to 8 are obviously wrong. Actual data backs you up. Guy. Thank you, Guy. Yes. That's what we've been saying all along. Yet, major media still calls it the more transmissible variant. Oh, well.
Oh, well. It may have a fitness advantage, but we don't know that it's
No, you
to
need
do with
to we
transmissibility.
need to do the experiments. And
By the way, Vincent's comment about Sophie's U.S. Open preferences was not entirely the non sequitur it sounded like to people who are seeing this only on audio. Sophie actually walked into the room and petted the cat. And
yes, that triggered my...
That triggered the question. It was nuclear power into the U.S. Open was otherwise kind of a...
Yeah, I guess so. I that's true. I didn't think about that.
It just occurred to me like, wait, people who are listening to this are going to wonder why the heck he's talking about tennis now. As a
teenager who is playing tennis, I thought she might be excited about that.
Yes, she's very, very keen on this.
Alan, could you take the next one?
Sure. Peter writes, you guys remain my heroes for the very best COVID information. Kudos and many, many, many thanks. I found the UK chart posted on the CNN website very interesting. I would like to see a similar chart for the US. It shows cases, deaths, and vaccination progress all together. I wish it would show masking, distancing, etc. as well, but I do know that the mid-July UK peak coincided with the relaxation of community preventive measures. From this, I conclude that greater than 50% community vaccination prevents deaths and probably hospitalizations. I also conclude that greater than 60% community vaccination does not prevent community cases. Some of these cases will be among the unvaccinated and some will come from the vaccinated. The dedicated fan learning tons about viruses, and Pete is directing the Office of Environmental Health and Safety at Yale. And this graph is very informative. It shows case counts and then against deaths, and you see the peaks early in the pandemic and the big post-holiday peak, and then the third wave in infections, and oh my God, we've got another peak in infections, but then you look at the deaths and the third wave is just this little tiny, barely detectable lump.
Yes, it's in a graphic way.
People are getting the virus, but they're not dying.
Yeah, it's great. I mean, that's what the vaccines are doing. I don't understand.
That's the whole point.
I don't understand
that is the whole point.
Well, the issue, I think, is that we got so primed to follow infection rates very appropriately early in the pandemic. It was a very important number to track.
Yeah.
And now, though, infection rates are being uncoupled in places that are highly vaccinated. They're being uncoupled from deaths and hospitalizations and bad outcomes. And that was the point of the vaccines. But now we need to kind of revise people's thinking on this. It should no longer be about obsessing over infection counts. We need to just chill on that. The issue is not infection. The issue is what's the infection doing?
That's right. Last night on PBS, they had an interview with the director of hospitals for the state of Idaho. And this guy was absolutely apoplectic. He had no answer for what's going on right now. And then Judy asked him whether or not all of the people that were sick had been vaccinated or not vaccinated. And he said, not a single one of the people that are on respirators were vaccinated, not one person.
Yeah. And
the hospital is now overflowing.
Yeah.
And it's not going to get better. It's going to get worse. So this is a repeat. We don't learn from history. We don't learn from our experiences.
Nope.
We are just dunderheaded. I don't know any other word to describe it.
No, I can think of some other words, but it's
I can too. But and then I heard another a sociologist say we tend to blame people for catching these viruses and getting sick because they haven't been vaccinated. We're shaming them, and we really shouldn't be doing that. We should be trying to find ways to get them to see why they should get vaccinated. Mm-hmm. That sounds like a good thing. But and then Tony Fauci was on last night, and he said. Judy said, well, isn't there something we can be doing about educating the public? He says, Judy Woodward, we've been doing this since the beginning of this outbreak. And every time we come up with a new take on it, we tell the people what to do. He says, and there's nothing simpler than going in and getting a vaccination. Nothing simpler. And he said, if you don't see the truth in that, then you have, not that you haven't been listening, you haven't been believing. It's not a question of have you heard this before or not. You just elect to just put your mind out in another place. And
we're running out of strategies. And I think at this point, a lot of us are running out of patience.
I think I think the licensure will make another surge. A lot of people have been waiting for that. So Pfizer, Moderna is upcoming.
It'll help some. It'll help with some of the people who are genuinely vaccine hesitant, but there is a significant population of folks who are just walled off in their disinformation bubble. And
100 million.
I don't know if all 100 million of them are actually unreachable, but an awful lot of them are unreachable.
I agree. Dixon, I agree that we should not shame them, but I think we shouldn't give up. So Fauci is implying that we've done a lot, and it hasn't. But I think we have to still try, and that's why we always talk about it on TWiV, hoping we reach one or more people.
That's right.
I don't think
it's the same message. It's exactly the same message. We have not veered from that at all.
By the way, the image, as for those of you who are just listening, it's in the show notes. But as Alan described, it's called "Vaccination in the UK Curb Third Wave Deaths," and the subtitle is: "By mid-June, when the COVID-19 cases surged again, 45% of Britons had been fully vaccinated, 62% had received at least one dose. The death toll remained relatively low." And the graph is just beautifully showing that.
Exactly.
And the graphs from Massachusetts look very similar.
Yeah.
We don't have a U.S. wide thing, and actually, I'm not sure it would be instructive because the vaccination rates vary so much by state.
Yes.
And even within states. But if you look at the statewide data, you'll see highly vaccinated states have this very similar type of curve where, cases come up and deaths not so much.
Right.
Dixon, can you take the next one?
Certainly. Owen writes, your listeners who play wind instruments might enjoy this Science News article on the science of keeping wind instruments from spreading the coronavirus. Now, I confess I have not looked at this, so perhaps one of you more informed individuals can tell the listening public what's going on.
Well, the article is called "What Science Tells Us About Reducing Coronavirus Spread from Wind Instruments." And they have a picture of a young lady playing it looks like a flugelhorn maybe.
Maybe I should look at this.
And she's wearing a mask and playing it. So a big trumpet, right?
She's wearing a mask.
Yeah, that's she looks like she's in a drum corps.
Yeah.
You mean there's a slit in the mask that allows the mouthpiece to go into her?
Oh, no, it's a high school marching band.
It doesn't look like it, Dixon. I don't know.
She's playing the instrument through the mask?
Looks like it.
Wow.
Anyway, this describes studies to track dispersion of air from instruments with mouthpieces and measuring the particles coming out and so forth, which kinds of winds pose the highest risk. And they have a graph here of aerosols generated by wind instruments. This is actually quite good.
This is useful.
The biggest, and this is measured in particles per liter of air. The biggest by far is the trumpet.
And the trumpet players are going to say, "Yeah, we win."
That's right, exactly right.
Players are.
And then the next one, which is over 10 times, about 10 times less. The oboe, the bass trombone, and the clarinet, they're all kind of similar. And then very low.
And the oboe is sitting in the back facing downward anyway, so it's not getting anywhere.
French horn, bass clarinet, flute, piccolo,
the tuba
bassoon.
is the safest.
Yeah, the tuba is the safest, generates very low aerosols.
Right.
So my friend Joe Terwilliger, if he's listening, Tuba Christmas should be able to go on as scheduled.
Absolutely.
They also talk about how your embouchure, remember that word, embouchure?
Who could forget? Yeah. I think about it all the time.
The way you put your mouth on the mouthpiece, how that influences the generation of aerosols. Right. They give you some safety tips. So for those of you, and they have a list here, masks and covering, distance, timing, 30 limit, rehearsal limit indoors, 60 minutes outdoors, clear the air between the next one.
Right.
Empty spit valves into buckets with alcohol solution.
Exactly.
Spit valves, yeah.
Anyway,
maybe the orchestra, if they're sitting, like the New York Philharmonic, for instance, maybe they should just all turn around and put their backs to the audience.
Well, the French horn players already kind of do that. They point the horn back.
That's right. That's true.
Just a badly designed instrument.
Anyway, those of you who have asked about this, check it out. I mean, these are instruments. It's not about singing. That's a different story.
And this is looking, this graph is looking at the aerosol production. It's not looking at the amount of virus coming out. It's certainly not looking at the infectious virus coming out.
Right.
And that gets back to something we talked about fairly early on in the pandemic about brass instruments versus woodwinds. brass is copper,
Yes.
which I would expect would do a number on the virus. And
It's true.
quite possibly even those trumpet players who are spitting out huge quantities of the stuff straight ahead of them, by the time it gets through the whole trumpet, I don't know how much virus is actually in there.
I actually don't know how you'd play a trumpet through a mask because
I don't think you can do it.
the skin is what.
There's no armature at that point.
No.
Right.
All right. CC writes, I initially found TWiV around December 2019 by serendipitously stumbling upon Vincent's virology resources. I was pleasantly surprised to see the podcast pick up once SARS-CoV-2 tumbled onto the scene, which has kept me both informed and entertained in the lab throughout my first year as a grad student in molecular microbiology. During TWiV 792, Vincent and Kathy were discussing the issue of public vaccine hesitancy. We do it in every episode, right? Vincent insists some of this hesitancy is politically driven, but Kathy responded she believes it is a historical human nature issue. I'm curious what Kathy means by this, as she didn't have the chance to explain her thinking. I think we need to ask, what gave rise to the vaccine hesitancy of the last year and a half? The three most pervasive moments of anti-vaccine sentiment in history lack continuity in that they emerged independently. The initial anti-vax movements of the 1800s arose as a reaction to compulsory vaccination. However, those groups died out long ago of smallpox. The anti-vaccine crowds of the late 90s and early 2000s arose in the wake of disgraced former physician Andrew Wakefield's attempt to defraud the public by fabricating data linking MMR combination vaccine with autism. Of course, the progeny of that moment are still around, but did they seed the current vaccine hesitancy of 2020-21? I'm skeptical of this because much of the sentiment against vaccines today has nothing to do with MMR or autism. Rather, I think the modern anti-vax movement has arisen out of the uniquely American phenomenon of stubbornly refusing to do things in the best interests of oneself and or others simply because government is the one issuing the policy recommendations. There's a large subset of the U.S. who think they are rugged individualists and fantasize about being part of some burgeoning movement that could one day overthrow a perceived tyrannical government. In reality, they function more as a reactionary collective id that can be easily drafted by ideological allies in politics or media, for example, Rand Paul, Tucker Carlson, Eric Weinstein, to spread misinformation and propaganda. I think this is demonstrated, at least in part, by the fact that the majority of misinformation disseminated to and recited by the anti-vaccine people is broadcast by such political channels rather than originating from some grassroots opposition or instinctual skepticism toward vaccines. Remember, the majority of these people were not vehemently opposed to vaccines or mandates prior to COVID-19. Most of them would just have their children complete the vaccination schedule as normal and move on. The current incarnation is largely the result of astroturfing and are ideologically homogeneous. On the upside, most of these people will just cave in and get vaccinated once the inconvenience of being unvaccinated outweighs the desire to stubbornly refuse it. I think history bears this out. For example, the inconvenience of passing through the TSA checkpoint is still outweighed by the desire to fly. Thanks for all you do to help make microbiology, immunology, and virology more digestible by the general public. Cheers to the next year of science and learning. Regards, CC. And thank you, CC. Wonderful letter. So well written.
Good one.
There is overlap, I think, between the MMR autism proponents and the ideological current wave, just because the same people were still around and they saw the opportunity and figured they'd continue exploiting it. But I think this nails a lot of what's going on.
I would agree.
Alan, can you take the next one?
Sure. Preben writes, Dear TWivers, I'm a veterinary immunologist teaching and researching at the Faculty of Veterinary Medicine outside Oslo in Norway. First of all, I want to give my wholehearted thanks for your tireless effort to inform deep and wide about everything viral. Since the pandemic outbreak, you've been my primary go-to source for updated information. I've been enlightened and entertained, and not the least, learned a lot of virus immunology, which often finds its way into my own tuition. Thanks also for your other podcasts, of which Immune is, of course, my favorite. I'd like to comment. This sounds like somebody who might be a good guest, actually. I'd like to comment on episode 791, where you discussed a study in rhesus macaque monkeys where T cells were depleted using antibodies, yet the monkeys recovered from COVID infection. Apparently, T cells should then not have been important for the recovery. I did not hear you mentioning resident memory T cells. These cells reside for a long time in different tissues, including in respiratory organs. Recent work shows that in mice, these T cells may accumulate and may eventually comprise as much as 25% of the cells in a given tissue, gives a reference. The same group of researchers have found that these cells may in fact be stimulated upon infection, recirculate, expand, and find their way back into the original tissue, gives another reference. In the macaque study, antibodies were shown to efficiently deplete circulating T cells, but to my best understanding, an antibody injection would not be able to reach all T cells residing deep inside the tissues. Thus, a large pool of resident T cells could have remained in these monkeys, been stimulated in the experimental infection, and contributed to disease protection. If so, the study cannot be a conclusive proof of T cells being dispensable for disease protection. Please keep up your tireless work and keep it auditive. I always listen to you while doing something else, and I really appreciate when you make some extra effort to explain what figures say. P.S. You keep saying that mice are not people. I do agree, so we try to bring mice into a more real-world situation by raising them in large natural habitats full of soil, animal manure, and other farmyard ingredients, and provides a link to that. So does the Massopost group mentioned above, whom we collaborate with. They let laboratory mice spend time with pet shop mice to bring in microbes from the outside world. Both of our groups find that naturalized mice acquire a more human-resembling immune traits. They are even better protected from colon cancer, to be published.
Wow.
Best regards, Preben, who is associate professor and head of the unit, the immunology unit at the Norwegian University of Life Sciences. Thank you very much.
That's cool. Yes, that's a great point of these tissue-resident T cells. Yeah, I think that's a great point. They didn't look at those in that study.
You can't be sure you got rid of all the T cells.
Yeah. Very nice. Dixon.
Gregory writes, Dear TWiV team, you've mentioned several times on the show that cheap home antigen tests never became a reality. Living in Germany, I've been somewhat puzzled whenever you guys say this. Here, a pack of 5 rapid antigen tests costs €3.75. This is cheap enough so that most people could afford to test themselves daily if they cared to do so. The other day, I looked up how much home antigen tests cost in the U.S. Much to my surprise, a 2-pack costs $25. I've tried to figure out why rapid antigen tests cost so much in the U.S. compared to Germany, but I can't find any explanation online. This seems like the kind of thing that Alan Dove would know about. If there's any explanation for the massive price difference, I'd be interested to know. Thanks for producing this great podcast. Best regards, Gregory. P.S. Here's a link where you can see prices for home antigen test kits in Germany. What do you think, Alan?
I say, welcome to America, where everything in medicine costs 10 times more than it does anywhere else in the world.
That's right.
Our prescription drugs, our doctor visits, our everything about our healthcare system is massively overpriced compared to everywhere else in the world. And
you're right.
That's
the way it is.
That's something we can't seem to change.
Do you think it's because they're subsidized in Germany?
It is. There's a thicket of problems here. There are subsidies. There are price restrictions. Other governments have the power to say, "Thou shalt not charge more than this amount," and thus it is. There are structural issues in our healthcare system that add costs. We've got to make sure we pay enough of a profit to the insurance companies to keep their shareholders happy. That's not the case in many other countries. This is our system.
So, and if the federal government put a limit on the price, the states would say, "To hell with you! I'm doing."
And gosh, I mean, price caps on things. Can you imagine politically how that's going to play? Unless it's a farm product, you can put a price limit. You can have a minimum price for milk to make sure that you prop up the dairy farmers, but you can't set a maximum price on a drug, because then you're tampering with the markets, and that's socialism, and we all know how bad that would be.
Absolutely.
My gosh. Gregory, enjoy your country.
Yes, Gregory. Yeah.
All right, one more from Jeff. Hey, on your last episode, you stated, while on a flight looking down at the ground, you wondered why rivers meander. Well, if you have an hour, and we already know you have a knack for critical thinking, this link should get you into the ballpark. This is another professor, in my opinion, that deserves a seat at the round table of science communication. He has an infectious teaching style, complete with all old school chalkboard discussions. He has an enthusiasm in his presentation that hooks you. If you were ever to have a roundtable discussion about science communication with other communicators from other disciplines, I would suggest him with others like Neil deGrasse Tyson. His lectures are called Geology of the Pacific Northwest with Nick Zentner, and also has a PBS series in the Seattle area. My first introduction to Nick was Super Volcanoes of the Pacific Northwest, and I have been hooked ever since, and I hope you enjoy and agree. And Jeff provides a YouTube link. Thanks, and as always, keep up the great communication. I guess it's
Cool.
Jay, because I put Jeff. It's not Jeff. It's Jay. Sorry, Jay. Oh, thank you. And I think Alan was not on that episode, or else you probably would have told us why rivers meander, right?
I don't know. I certainly can't give you the kind of depth that I think this video goes into. I only started watching it. It's off to a good start. The old saying I've heard is that following the path of least resistance makes men and rivers crooked.
That's great.
So it's about the hydrodynamics of it, and beyond that, you should go and listen.
I'll watch it because I want to know. Dixon, do you know why rivers meander?
Well, it's funny you should ask. How much
Dixon
time
is
do
gonna
you
meander
have?
through an explanation.
No. I'm not going to meander at all. I'm going to recommend another resource, and that is the son of Aldo Leopold, whose name was Luna Leopold. And he taught at Harvard University, of all places. And I actually know someone who took a course from him. And he was an expert on the physics of river meanders. And there's a mathematical formula for this. The wider the river, the regularity of the meanders changes as the width of the river changes. So it has something to do with the velocity of the river as well as the substrate that it's running over. But if you fly over the south central part of the United States and you're looking at the Mississippi River meandering, you can't figure it out. It just doesn't look like it makes any sense whatsoever. It's got so many twists and turns in it. It looks like an anaconda that swallowed itself. And a number of I'm blocking on the name of these lakes that are created by Oxbow lakes that are cut off by the currents as flooding occurs and then fills them in. It's amazing to see how nature has its way with the landscape. But that having been said, Luna Leopold is another authority that I highly recommend people go to if they want to know something more about I know the mathematics of this is going to probably be over a lot of people's heads, including mine. I've read some of his work. It's fascinating stuff to realize that there's a science to this. It's not a random walk. Let's just put it that way. It's not a random walk. And to have a father like Aldo Leopold must have been incredibly inspiring to a kid who eventually ended up studying the way rivers behave. It's just an amazing connection there. So I'm amazed. That's enough I have to say about it. But it's a fascinating subject. It really is.
Of course, Dixon has thelivingriver.com where you can find out about rivers as well.
Indeed. Yeah, it doesn't cover much about meandering, though.
No, it doesn't.
Well, I think the purpose of a meander from that perspective is to provide trout habitat, right?
Exactly. I meander a lot down those rivers, and I will again, I'm sure. So, meandering is a river characteristic.
All right, time for some picks of the week. Dixon, start us off.
Okay, so, continuing on with the humor theme of last week, where I picked a Victor Borge skit about punctuated language, I thought, who are the great humorists out there that really everybody will recognize the universality of their humor? You don't have to be of a political persuasion or of an age group to realize how clever these people are. And one of them stands out of almost all of them. And he's the official Droodles inventor and also invented Mad Libs. And if you'll ask me what his name is right now, I'm just blocking like hell on it. But
Roger Price.
Roger Price. And Roger Price was a joke writer for Bob Hope. His bio is in this link that I've put into the show notes. And he was an amazing person. He would go on stage at, let's say, the Village Vanguard or some of those nightclubs down in New York City in the early 60s, and he would take out an easel. He would put up a piece of blank paper. He would tape it to the top of the easel, and then he would take a pen and he would make a straight line right across. He said, I thought I would want to show you some of my recollections of the landscape of Texas on my last vacation. He would do visuals like, I'm going to do this for the three of us, but you can't see this at home because I guess maybe you're not watching, but you take your index finger and take all the other fingers of your other hand and make a ball on the top of the finger. And then he would say, what is this? And what is this? Who the heck would ever know? And he says, oh, that's a worm with a shampoo. That was the level of his humor. He was so clever. He had one showing a circle with 8 spikes coming out of it, except for one of the spikes was straight down. Okay, and it's too bad that we couldn't have talked about that to Maya, because what that is a depiction of a spider doing a push-up. Or a handstand, sorry, a handstand.
Handstand, yes.
A spider doing a handstand. Sorry.
Yeah, and this was big in the 50s, but there's a more recent use of one of them, Ship Arriving Too Late.
Yes,
which
Frank
was
Zappa.
The title of a Frank Zappa album.
Exactly.
And it was the album on which the song Valley Girl was recorded.
Wow.
Yeah, he was incredibly inventive and poorly understood, but had a following that was absolutely, he had his own TV show for a while. I used to watch it all the time. He's in the same category as Ernie Kovacs. He's like an abstract humorist.
Yes.
And I just love his take
Classic.
on life.
These are fabulous. I'd never seen this before, actually.
Oh,
And
really?
I love there's a scene in Texas. It's just a horizontal line.
That's exactly right. So I had a thought while I was looking at all these droodles.
Wow.
What if you took some of the features of a capsid of viruses, various viruses. Each one has a different set.
Come up with
you
a
abstracted them and then used them as flashcards and said, which virus does this remind you of? And you could actually teach a little bit of virology this way if you wanted to.
Yeah. That's great. By the way, Dixon's site is thelivingliver.org.
Yes.
I keep saying .com, but it's .org. And it's really a wonderful resource. For example, you've got, river types.
Yeah.
Freestone, feeder stream, limestone stream, spring creek, tailwater fisheries, energy flow, galleries, glossary, appendices. It's a really good site, Dixon.
Thank you, Vincent. It wouldn't have been up unless you had helped me greatly with it.
That was just mechanical. You did the content. It's really good.
Well,
and look at the galleries. The photos are gorgeous.
Photos are great.
Thanks so much.
The only thing missing is meandering rivers, but that's not your thing.
This is true. It's
a meandering site about rivers.
It is.
There you go.
Alan, what do you have for us?
I have the most important scientific conference of the year. That just happened. It had to be online again this year, like so many things. This is the 31st annual Ig Nobel Prize ceremony. And you can, because it was online, you can watch a video of it. They also have the text giving a rundown of what transpired at the ceremony. And this first person that I see here, he's a takeoff on Harpo Marx. Is that the idea? Mark Abrams would very much appreciate the comparison there. He's the guy behind the Ig Nobel Prizes. Mark has been doing this. He runs the Annals of Improbable Research. So he's the host. Nominally, this incredible zaniness that normally happens in a theater at Harvard University, but they had to do it online. And they actually did a really good job, I think, with using the medium of Zoom video to its fullest potential.
Great. And how does this compare with the Darwin Awards?
This is a much more extensive and long-standing tradition.
Okay.
Someone asked me recently, what is an Ig Nobel? And I couldn't describe it, but here it says...
It is a prize for science that makes you laugh and then think.
Yeah.
Okay.
It's not necessarily for things that are wrong, right?
No, it's not. It's not.
Okay, so the Darwin Awards are different then.
The Darwin Awards are, for people removing themselves from the gene pool. But
exactly.
The Ig Nobel prizes are for things that are science or could be defined as such, and yet somehow seem really funny.
So, for example, the Ecology Prize goes to people for using genetic analysis to identify the different species of bacteria that reside in wads of chewing gum stuck to pavements in various countries.
Yes.
And the name of the paper is the Wasted Chewing Gum Bacteriome. So that's an example. That's great. We have biology, chemistry, medicine, for demonstrating that sexual orgasms can be as effective as decongestant medicines at improving nasal breathing. Oh my gosh.
Really? Really?
Yeah, these are wonderful. Wonderful.
Yeah.
I guess the real Nobels are coming up soon, right?
Yes, there's an autumn thing as well.
October, that's right.
Do you think
Nobels, and stock market crashes? We do those in the fall.
All right, my pick is I have a New York Times article on Biden's announcement yesterday to do federal and business vaccine mandates, right?
Yes.
U.S. government, all federal employees, federal contractors have to be vaccinated. Already, nursing home workers that treat patients on Medicare and Medicaid already have to be. He's used OSHA to make employers with 100 or more employees require that they be vaccinated or else get a test a week. And many big companies are already doing this.
$25 for 2 shots.
Many big companies are already doing that. United Airlines, Disney, Fox News. It's interesting that they require that. Then on the other hand, they trash vaccination. Yeah.
Wow.
100 million Americans should be affected by this. 2/3 of all U.S. workers. So many of them are probably already vaccinated, right?
Yeah.
Also, he urged large entertainment venues to require vaccines or show a recent negative test. Of course, here in New York City, when Broadway opens, you're going to require proof of vaccination, I believe. Many restaurants already do that. Already has required teachers at schools run by the Defense Department to be vaccinated. Today, the government will require teachers in Head Start to be vaccinated. And he wants governors to require that all teachers and staff be vaccinated. Wow, let's see how that plays out. They're going to be legal challenges, I'm sure.
It's going to play out exactly as you would expect it to be.
That's right.
He's using the Defense Production Act to increase production of rapid tests.
Right.
The government has expanded free testing at 10,000 pharmacies. Will spend $2 billion to distribute 300 million rapid tests to community health centers, food banks, and schools, and told the TSA to double fines on travelers that refuse to mask. I often see travelers on trains and subways and who don't have a mask. People say, put it on, and they put it on. I've never seen anyone refuse, but I'm sure there are some. Anyway, I think this is going to be interesting.
Very.
As you may know, many years ago, the Supreme Court did say it was legal to mandate vaccination. So I guess that's going to be challenged. We have a listener pick from Anonymous. I am just a humble molecular biologist on the job market who is reluctant to share current niche meteorological realities owing to any ire I might provoke with this request. A friend recently shared a non-science podcast with me that did a deep dive on recent ivermectin retractions. And it's called Behind the Bastards and links to a YouTube channel. In addition to being humorous, the hosts and guests enumerated problems with preprints, meta-analyses that include outliers and or studies that are later retracted, and conflicts of interest. The host also named serial retraction offenders, for example, Pierre Corey, MD, and cited articles and journals regarding retractions, revisions, and removals. The cited journals don't take a stand on ivermectin, but they do make clear their position on studies lacking appropriate controls and studies based on fraudulent data. To be clear, the aforementioned journals did the very least they could do. A couple of takeaways from all the above are outrageous to me, and I hope you all can use your platforms to thwart these in the future. Number one, 3 published meta-analyses on ivermectin for COVID included a preprint study out of Egypt that was not yet peer-reviewed and later deemed fraudulent and removed from the preprint server. Research Square preprint server link below. Meta-analyses should never be allowed to include studies that are not peer-reviewed. Biostatistician, let me say that again. Biostatisticians stay way too busy flooding journals with reanalyses to pad their CVs, in my humble opinion. And this unscrupulous fame-seeking decision by 3 independent publication-hungry groups may have had devastating global consequences. How each elected to do this in 2021 after the last year we had is completely beyond the pale. Additional PSA, please tell your students and postdocs only to cite primary research articles in their dissertations and manuscripts. For example, do not cite review articles without primary data backup. Number 2, rather than being buried at the end of a publication or preprint, conflicts of interest, funding sources, and maybe previous retractions for authors should be listed right under author affiliations. I feel this should be especially true on preprint servers like bioRxiv, a Chan Zuckerberg joint, by the way, but why not on PubMed? To illustrate the point, Pierre Corey, MD, made a statement to the Senate claiming ivermectin is a miracle drug for COVID-19, link below. Corey is listed as the first author of a biostatistical meta-analysis of ivermectin studies published in the American Journal of Therapeutics that includes the debunked Egypt study that was removed from the preprint server above. This Corey et al. AJT article has been cited 37 times at the time of this writing, and A, includes no correction notice indicating the inclusion of the flim-flam Egypt study. B, lists no conflicts of interest for Corey, who heads the Frontline COVID-19 Critical Care Alliance, also allied with a British ivermectin recommendation development group comprised of self-proclaimed, highly publicized, world-renowned critical care physicians and scholars. It links below. C, is the only meta-analysis or biostatistical paper whatsoever to be authored by Corey, who is a MD with no record of publication in biostatistics. D, was rejected by Frontiers in Pharmacology on the basis of unsupported claims after further review following provisional acceptance, later published in AJT. And number 3, not for nothing, of Corey's 51 publications currently in PubMed, A, one article title includes the word corrigendum, typo, not a COVID paper. B, one article title includes the word reply. C, 2 article titles include the word rebuttal. D, 2 article titles include the word counterpoint. D, 5 article titles end in a question mark. E, 5 articles are case studies, single patient. 6 are editorial articles. 8 are comment articles. 11 are review articles, cherry-picked. 19 include a total of 3 or fewer authors, including Corey. 6 include a total of 2 or fewer authors, including Corey. Corey is a busy MD who knows how to juke the publish or perish system and who also doesn't have a lot of peers in the primary research community. Only quantity, not quality, for this highly published, world-renowned critical care physician for those who didn't hear it twice the first time. In closing, I sincerely apologize for the length of this rant, but if you don't want to wade into the troubles with meta-analyses and preprints, I hope you can at least clue listeners into the Beyond the Bastards pod on ivermectin and maybe mention Corey's troubling involvement. The worst thing that I can imagine is that ivermectin is actually helpful for some subset of the population at some stage of its interactions with SARS-CoV-2. But none of the researchers who can best elucidate this possibility will get support to do so because quacks, unscrupulous and publication-hungry statisticians, and shite journal editors have so sullied the field. With gratitude and esteem, Anonymous. And following are all the links mentioned, which you can find in the show notes. And Anonymous, it's an amazing letter. Thank you so much for going through this.
I must say that I ran across a reference to this data, in quotes, unquotes, in Yahoo News. And the headline read, Nobel laureate approves of the use of ivermectin for treatment of COVID-19. And it was none other than Bill Campbell. And Bill Campbell, in the beginning, when he was told this first, said, gee, wouldn't it be wonderful if ivermectin did have some effect against the COVID virus? But then he took it back as soon as he found out that it didn't. And he said, no, I never said that it did. I said, wouldn't it be nice if it did? And then, of course, that's how they blew this thing up into a headline that convinces people that knowledgeable people think this is good.
Well,
sham.
Now, Anonymous, you were sad that maybe the right studies will not happen. But in fact, there's a large controlled clinical study underway,
That's right, there was.
as Daniel Griffin has mentioned. So hopefully we'll get a final answer.
It's crazy.
When I was on the Lex Friedman podcast, he asked me about ivermectin. We talked about it, and I mentioned an anecdote that a few of our friends, vaccinated people, got COVID, and one of them got very ill. He had many other comorbidities, was hospitalized, and then was automatically given ivermectin, hydroxychloroquine, and dexamethasone, and then the next day remdesivir. And they told me that's the standard of care in many hospitals in New Jersey. And so one of our listeners wrote in and said, someone could take that clip from Lex Friedman and have you saying it's the standard of care in New Jersey and make you look like
Tape
you're
it together, and that's right. That's
endorsing it.
Sure, people do that all the time. What can I do? I can't really control what I say. But as Daniel Griffin has mentioned many times, there's no similarity in what's done in his place and somewhere else. Just
Vincent, I would like to correct what you just said, because you can control what you say, and you do that very well. What you can't control is what you say from other people saying what you say.
Well, remember the interview with Peter Daszak in Singapore.
Yeah.
Fox News took a clip and said this shows that he made the virus.
Of course.
That's crazy, right? I try to be careful about what I say, yes.
You're very careful about what you say.
All right, TWiV 804, show notes, microbe.tv/TWiV. Send your questions and comments. We love them. We do love them. TWiV@microbe.tv. If you like what we do, please consider supporting us financially, microbe.tv/contribute. Dixon Despommier is at Racaniello.org, thelivingriver.org. Thank you, Dixon.
You're welcome, Vincent. And it was kind of interesting and nice to have a smaller group this time, because I guess we got a lot of better chances to say what we felt about subjects that don't necessarily relate to virology. And it was great. The guest was wonderful. I really enjoyed that very much.
She was awesome.
Yeah,
Thank you so much for having her on.
Yep. Alan Dove is at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thanks. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.